Good day, this is Professor Will, CBMD PhD. Today's Saturday, March 27, 2021. It is 2:44 and 48 seconds p.m. Eastern Standard Time. Today's research, cell signals. So let's hop to it here. Eat me, the cell signal of death. An eat me signal displayed on cell surfaces requires activation of a lipid scrambling protein by a nuclear protein fragment. Date March 26, 2021. Source, Kyoto University. Summary. Scientists have revealed molecular mechanisms involved in eliminating unwanted cells in the body. A nuclear protein fragment released into the cytoplasm activates a plasma membrane protein to display a lipid on the cell surface, signaling other cells to get rid of it. Every day, 10 billion cells die and are engulfed by blood cells called phagocytes. If this didn't happen, then cells would burst, triggering an autoimmune reaction. It is important to understand how dead cells are eliminated as part of our body's maintenance. Scientists already know that dead cells display an eat me signal on their surface that is recognized by phagocytes. During this process, lipids are flipped between the inner and outer parts of the cell membrane via a variety of proteins called scramblases. Researchers have already identified several of these lipid scrambling proteins, but some of their activation mechanisms have been unclear. To solve this, the team used an array of screening approaches to study the scrambling protein called XKR4. The broad aim was to single out the genes that are active during cell death and to specifically zoom in on XKR4 and its associated proteins to understand how they interact. We found that a nuclear protein fragment activates XKR4 to display the eat me signal to phagocytes. Specifically, the scientists found that cell death signals lead to a nuclear protein called XRCC4 getting cut by an enzyme. A fragment of XRCC4 leaves the nucleus, activating XKR4, which forms a dimer, the linking of identical pieces into configurations. Both XRCC4 binding and dimer formation are necessary for XKR4 to ultimately transfer lipids on the cell surface to alert phagocytes. XR4 is only one of the scrambling proteins. Others are activated much faster during cell death. The team now wants to understand when and why the XKR4 pathway is specifically activated. Since it is strongly expressed in the brain, it is likely important for brain function. Researchers are now studying the elimination of unwanted cells or compartments in the brain to understand this process further. Good day, this is Professor Will, CBMD PhD, on microbiology, bioinformatics, and pathophysiology. This video is strictly made for educational purposes only. Well, hope you enjoyed listening. Until next time, have a great day.